So we are module 11, page 3. Last week we introduced and then finished with regards to the stages of coagulation. And really what we're talking about is blood as a connective tissue. But in terms of this hemostasis process, and that is the process whereby blood clots ultimately. There are 3 stages. We introduced the vasoconstriction phase, and that's just after a vessel of any kind ruptures. The vessel in particular constricts, and then we talked about the platelet plug stage, and in terms of damaged vessels, there's a positive feedback mechanism that causes blood platelets to swell and get sticky and stick to both the vessel and one another. And that's what we're talking about in terms of both the clot that you don't see and the clot that you do see in the form of a scab on the exterior of your skin. So again, what's going on there is an aspect of the stopping of the blood loss. And we also referred to a negative aspect of this platelet plug stage, which is the buildup of those things in your vascular system, ultimately to possibly break loose and cause issues in your brain in the form of a stroke or even in your heart. And so this morning we're beginning the next stage of this, and that is called coagulation. So when a tissue or vessel is damaged, the chemicals in the bloodstream, I'm on page 3 near the top, the chemicals in the bloodstream react, setting off this amazing cascade of chemical reactions which ultimately produce a number of these different things, and I wanted to refer you to that. Let's see here. Okie dokie. And no wonder, to help if I was on the right module. There we go. So I haven't asked you to know figure 11.7 on page 329. What I would like you to be familiar with is what is in your notes and what I'm going to read to you now. So just to be familiar with this. So this is the process. Prothrombinase is an enzyme that works on prothrombin, and this is the stage 1 of coagulation. Prothrombin is always in the blood of a healthy person. Prothrombinase converts that prothrombin into an enzyme, thrombin. This is stage 2 of coagulation. Stage 3 is thrombin interacting with fibrinogen. That's one of the proteins in blood plasma, which is water soluble. It's converted into fibrin, which is no longer water soluble, and forms instead large lone fibers that form a net for trapping other components of blood inside the vessels. These 3 steps that lead to blood coagulation occur because of coagulation factors. And those are proteins in blood plasma which help initiate the blood coagulation process and are manufactured in the liver. They require vitamin K, the result of vitamin K from diet, and from bacteria, interestingly enough, which live in our intestines. Hemostasis is stopped. Our blood doesn't continue to coagulate after a wound because of a body process, anticoagulation. And what happens there is anticoagulants prevent certain blood factors from being activated. So that's how you stop blood loss, and then once it's stopped and the healing of the vessel takes place, the coagulation stops, and we're back to good. So there's a longer discussion of this in your textbook, which you might be interested in reading, but what I'm interested in you being familiar with is what I've given you in your notes. All right? So is there any questions about that process? Okie dokie, let's talk about blood types. Always an interesting aspect, I think. Of course, there are four, this is page 332, introduces the discussion, and there is a handy-dandy little figure there on page 333, which very well might help you in terms of grasping what we're going to talk about. Again, the pictures always help me. There's four blood types. So A, B, and O. All right, so A, B, and O. The blood types are differentiated by the antigen molecules that they contain or they don't contain. An antigen is a protein or carbohydrate that, when introduced in the blood, trigger the production of an antibody. And that's an immune response that fights off invaders. So the blood types are distinguished again by these antigens or not, and we'll explain. So type A blood has A antigens on their erythrocytes. Type B blood has B antigens on their erythrocytes. AB has both. It has both of those antigens. O doesn't have any. All right? So again, the way that these things are distinguished is what type of antigens do they have? A blood has A antigens. B blood has B antigens. AB has both, and O has none. And so again, in terms of the description in the bottom, what we're going to discuss too is in terms of what works with what. All right? So type A blood does not produce antibodies against A antigens. It does produce antibodies against B antigens. So in terms of you being able to receive B blood when you're type A, well, you're going to be in trouble because your type A blood produces antibodies against that type of blood. Type B blood does not produce antibodies against B antigens. It does produce antibodies against A antigens. And so likewise, if you were mistakenly given type A blood when you're type B, you got trouble. All right? Type AB blood does not produce antibodies against either one. And that would make sense, right? Because if it did, you'd be producing antibodies against yourself. And then finally, type O blood produces antibodies against both A and B, all right? And again, that makes sense. They in terms of the antigens on their erythrocytes, they don't have any. And so they're going to produce antibodies against anything, in essence. All right? So AB, though, will not produce antibodies against either A or B. So as it turns out, it isn't exactly known how antibodies against A and B antigens develop. A theory is that bacteria that carry the A or B antigen produce the antibodies. So, in other words, that process could go on in utero, or perhaps it's directly from mom, but it's because occasionally you'll have babies born with different blood types than mom, so it's just an interesting process there. Type O blood is called the universal donor type. O, A, B, or AB types have no types of antibodies against O. So that makes sense, right? However, the donor O blood does have antibodies against A and B, so they will attack the red blood cells of the host, but since you're not giving, at least most of the time, a lot of that type O blood, you're going to be okay. So in terms of the total blood volume. So not in sufficient percentages to cause problems as long as the blood donation is not great. The best donor, as you can well understand, is the one of the same type. So, and that obviously makes sense. So turn over to page 4 on your notes. This is the interesting discussion of genetics. In a basic biology course, you probably saw a little of this yourself in terms of the type here. So genetics determines our blood type. Type O blood is recessive, and so the alleles for A and B are both dominant over O. Types A and B are not dominant over each other. So AO alleles will produce A blood, BO alleles will produce B blood, OO alleles ultimately produce O blood. So in terms of the recessive nature of the blood and how it works in terms of the genotype there, that's illustrated, well, I've given you the same little example from page 334 as in your notes. The other aspect of blood typing, as you are aware of, if you've done your own blood type, yes sir, yes sir.
Then where does this idea of B positive and B negative come from?
You're right on track. That's where we're going next. Right now. It's called the rhesus factor. And so that is the other aspect of blood. So very good, very good. Look at the bulleted point underneath your little graph there in terms of the genotype. And that, you know, I couldn't remember the name, I just saw it. Punnett square. I was struggling. The hard drive was spinning and it wasn't going anywhere, but there it is. It's Punnett square, that's what it's called. But anyway, so the Rh factor stands for rhesus monkey, in which the Rh antigen was first identified. If the Rh antigen exists on the surface of erythrocytes, the blood type is called Rh positive. And if there is no Rh antigen, then it's simply called Rh negative. Some 85% of people are Rh positive, and it simply indicates the addition of that Rh antigen in your blood, in addition to whatever else you might have. Again, A-type blood with an A antigen and so on, except for O, which has none. Rh is dominant. Rh positive is dominant. So Rh plus R minus or negative alleles, you're going to be Rh positive. Only Rh minus alleles result in Rh minus blood. So in terms of the Punnett square, if you set it up for Rh plus and Rh minus, the only way you're gonna get minus is 25% of the time based on 2 Rh minus alleles. Rh positive blood will not produce antibodies against the Rh antigen, and again, that makes sense, right? You don't want to produce antibodies against yourself, since the body will not produce an antibody that attacks its own blood. Rh minus can produce the antibody against the Rh antigen. The only way an Rh minus person produces the Rh antibody is to be exposed to Rh positive blood. So, interestingly enough, the first transfusion of Rh positive blood for someone who is Rh negative, for the most part, won't hurt them because they have not had exposure to it in order to produce it. However, any Rh positive transfusions thereafter will cause a very bad reaction. Now, there's exceptions to this. A child or even an adult that never received a blood transfusion and mistakenly that is Rh minus, and mistakenly is given Rh positive blood, the first time could very well have a bad reaction because that child could have been exposed to his or her mother's blood if his or her mother was Rh positive in utero. So there is the potential for that. So, you never want to just say, oh, well, it don't matter the first time. It always matters. You always want to be certain about blood transfusions. And I might add, you'd think that's a simple thing. You'd think that with regards to modern technology and blood collection techniques, I mean, how we haven't perfected it, but it's so wonderful now to be able to provide blood for people who need it, and many people need blood. It's a good thing to give from time to time if you're physically able. They talk about that a lot, but it is a blessing. It's certainly a blessing. But the point is, you just can't ever take that for granted, and every year people are affected by receiving wrong-typed blood. All right, questions about that? In terms of blood type and the various antigens. I like the little drawing there on page 333. It's just kind of helpful for me to keep track of stuff. So let's talk about blood circulation. Blood circulation. On page 337, that's an overview of this thing. And kind of a nice little overview to describe direction, literally flow, and what's going on. So we'll first do a little bit of anatomy with regards to both of them. So I'm reading from blood circulation in the middle of page 4 of your notes. In humans, blood travels in a closed circulatory system. The blood is enclosed in blood vessels. Interestingly enough, arthropods have an open circulatory system. Just can hardly imagine how that works, but it does. There's 3 types of vessels, and you're well aware of all of them. There's arteries, capillaries, and veins, and that's more or less the flow direction is arteries to capillaries to veins. Arteries are blood vessels that carry blood away from the heart. A lot of times we think of arteries as being those vessels that carry oxygenated blood. Well, you might say they carry a higher percentage of oxygenated blood, but the veins will carry oxygenated blood too. It won't be depleted by virtue of it traveling through yourself before it returns to the veins. There's obviously still plenty of oxygen in your veins at that point in time. Capillaries are tiny, thin-walled blood vessels that allow exchange of gases and nutrients between the blood and the cells. The erythrocytes may squeeze through in a single file. Remember, we described that double convex, convex. Help the boy. Which is which again? It's concave, isn't it? Concave is the one that goes in. Double concave, I think. Oh, somebody look it up, so I'm not terribly embarrassed. We just talked about it. There's a few, where did it go? Let's look at it. Let's go back in time, back to page 319, and there it is. And I still don't see it. I see it, but I can't remember the term. Concave? Thank you. You helped the boy. It is concave. It's a cave. It's a little cave. And I remember how those things can fold over, and so they can fit through those capillaries. Just a remarkable thing. in terms of going through single file. What a remarkable thing, both the blood vessel and the blood cell. And then finally, veins are blood vessels that carry blood back to the heart. So in terms of that, again, there's oxygenated blood going through the whole system, but veins are heading blood back in the direction of the heart. There are two divisions to blood circulation. There is pulmonary circulation, and this makes sense, right? You have to get blood from the heart, which is the squeezing, the pumping station, to the lungs in order to fill that blood up with oxygen and then send it throughout the rest of the body. So there is pulmonary circulation of the blood over the air sacs in the lungs. So from heart to the lungs and back to the heart again. So that's what happens. You squeeze, you get movement to the lungs. The blood is oxygenated. It's sent back to the heart to get ready to be sent throughout the body. And then there is the systemic circulation, and that's circulation of the blood through the other tissues of the body where it delivers that oxygen and nutrients, and then picks up the carbon dioxide and the waste products by virtue of the use from the cells. The deoxygenated blood is dark red. It's often portrayed as blue, and it returns to the right side of the heart and pumps it into the lungs. The left side of the heart receives the oxygenated blood from the lungs and pumps it out to the tissues of the body. And that's what you see depicted there in terms of the drawing. And notice the obvious statement, but you know what? I confess from time to time it still confuses me. But the left side of the heart, as you're looking at it, is your right side, all right? Because you're looking at it, all right? And the way that you determine left and right is your left and your right, all right? So your right is the right side of your heart, and your left is the left side of the heart. So in terms of that, you see what's going on there, the pumping out from the right side of the heart and the pumping to the lungs from the left side. All right, excuse me, from the right side. See, I just did it. Look at the picture, and I knew I was saying it wrong. Anyway, on the right side to the lungs, and the left side to the body. So let's look at the anatomy of this thing, page 339. So not a bad idea either, page 339, figure 1110, or 340, figure 1111. Not a bad idea to draw those. Hint, hint. Not a bad idea. Again, that's how learning is done. At least one wonderful aspect of learning is drawing it. And I give extra credit for it, number one. And number two, you know, you don't have to be Rembrandt. You know, we don't have to have a precise drawing of this thing, but anything helps. Tracing even works. Y'all still trace? Is that something that you still do? Do you? All right. That's the way things got done when I was little. You didn't have copy machines, you didn't do any of that stuff, you just traced. All right, external heart anatomy. Heart, of course, consists of 4 chambers, 2 atria and auricle, and then 2 ventricles, and then, well, we'll just describe the rest of it. Let's start with the bulleted points. There's 2 atria or atrium. That's another word for entrance. Easy way to remember it. Entrances to the heart. They have thinner walls. Each atria has an auricle, a flap-like extension. At one time, the auricle meant the entire atrium. So in terms of terminology, that's changed. Actually, when I was young in my first anatomy physiology class, the auricles, the atria were described as auricles, so not anymore. So the auricle is the flap-like extension. And then the two ventricles. The ventricle means belly, on the belly, and they are the bellies of the heart. They have thick walls, and again, that makes sense because blood is entering through and into the heart with the auricles, with the atria, excuse me, there I go. And so they don't need to be as big or as muscular, but you think about what needs to happen in order to propel that blood out of the left side of your heart and get it to your feet and back. All right, and of course it's helped by movement and these sorts of things as well. But you know, you got to have a pretty muscular little doodad there in order to perform that. With regards to the atria, however, we're just talking about moving from side to side, and even to get the blood up to the lungs, you know, we need something substantive in order to do that, and that's the ventricle. Several large veins bring blood into the heart. So the superior vena cava brings blood from the upper body tissues to the heart. So in terms of these things, you see them labeled there, and again, it's helpful to sort of grasp them. And again, I think we're going to be on track to do our dissecting our cow heart and cow eye. I think we're going to be okay to do those, and we will attempt to look at these things. Sometimes they're clearer than at other times, but you see the superior vena cava there depicted both on the anterior and posterior view. I'm on page 339, the drawings there. You see the inferior vena cava as well. It is depicted at lower in terms of the same purple color, but the inferior vena cava is bringing blood from the lower body to the tissues of the heart. So both of those things make sense. The four pulmonary veins, the two left pulmonary veins and two right pulmonary veins, bring blood from the lungs into the heart. Now, again, I made the distinction a moment ago to tell you that sometimes we think of arteries as the oxygen delivering large vessels. And I mentioned to you that no, in fact, both of them, both veins and arteries, deliver oxygen in a greater content for the most part in arteries. But notice the subtle difference here. Remember, the definition of veins is bringing blood to the heart. And so the blood coming out of the lungs is delivered in a vein as it's going to the heart. And coming out of the lungs, we understand that blood to be as richly oxygenated as any blood in the body. So in this instance, we've got a vein delivering the blood with the greatest concentration of oxygen in all of the body. So let's see. Where were we? Okay, yeah, the four pulmonary veins. Two left, coming from the left lungs, and two right bring blood from the lungs to the heart. Two arteries carry blood away from the heart, the pulmonary trunk. Again, that's there's the pulmonary arteries there depicted right in the middle, second one down from the aorta. They carry blood out of the heart. The pulmonary trunk carries blood out of the heart to the lungs and quickly divides right and left to the right and left lungs. The aorta carries blood that is being sent to all other tissues. And so you see that great big aorta there depicted top middle. If you would go to the next page of your notes, page 5, internal heart anatomy. And so if you would turn over to page 340, and there is a pretty little drawing that I will refer you to, and we will again attempt to look at some of this stuff when we dissect. So the right atrium connects with the inferior vena cava and the superior vena cava and the coronary sinus the major vein coming from the heart tissue itself. So in terms of this, we see the right atrium depicted and what we're talking about here. The right atrium leads to the right ventricle through the right atrioventricular canal. And again, I refer it to you because you saw I already was confused this morning. When I'm looking at my left and right, let's see, okay, that's right. So what we're talking about is on the right side of you. It's on the left side of the picture. So in terms of, let me read it, start it again. The right atrium leads to the right ventricle through the right atrioventricular canal. The right atrioventricular valve or tricuspid valve is made of 3 layers of thin connective tissue, each of which is called a cusp, and it helps to regulate how blood flows through the canal and is passively controlled on pressure differences when the ventricle contracts and relaxes. Papillary muscles connect to the valve via the chordae tendineae or heartstrings. Yeah, and we will actually look at those heartstrings, which contract when the ventricles contract to keep the valve, more or less, from blowing out into the atrium, allowing all the blood to go to the pulmonary tract. So after blood has flown into the ventricle, it contracts or squeezes, and you can envision blood being forced out of that ventricle and headed in the direction of the right and left lungs. The left atrium receives oxygenated blood from 4 veins and is separated from the left ventricle by the left atrioventricular valve. That, as it turns out, is a bicuspid valve made of 2 sheets of connective tissue and opens in response again to pressure changes. The blood is flowing down the left atrioventricular canal to the left ventricle, which then opens into the aorta. So you receive the blood from the lungs, it goes into the left atrium. The left atrium opens into the left ventricle. When the left ventricle squeezes, the pressure changes, then of course allow the process to continue, and of course that blood is sent from the heart to the various parts of the body. The intraventricular septum is a muscular wall that separates the left and right ventricles, and you see it there depicted in the yellow. You see that massive muscular piece there in terms of that muscular division. The aorta and the pulmonary trunk are separated from their ventricles by the semilunar valves. Those are half-moon-shaped cusps of tissue that prevent backflow into the ventricles. The valves open passively due to blood flow out of the ventricles and are one way, closing off the ventricles to backflow. So in terms of preventing, backflow is the best word I can give you, of blood back into the heart, that's how those valves are working there. And in terms of all of this, what I've tried to do for you is digitally describe blood flow. Turn over to page 343 and you see that depicted in terms of that drawing. And what I've done is more or less tried to describe that for you digitally in this middle bulleted point there on page 5. So if you want to follow it in terms of the picture on page 343, I'll read it from the words on page 5 of your notes. So, deoxygenated blood back to the heart through the veins, the inferior vena cava, the superior vena cava, and the coronary sinus bring blood into the right atrium, which contracts, sends that through the tricuspid valve into the right ventricle. That right ventricle contracts or squeezes. It goes through the pulmonary semilunar valve into the pulmonary trunk, that vessel, one of two of those pulmonary arteries. The blood goes to the lungs, where it's oxygenated, and you drop off the carbon dioxide. The pulmonary capillaries, that's where that stuff happens. Then to the pulmonary veins. Then to the left atrium, which contracts. It opens that bicuspid valve, so that allows the blood to flow into the left ventricle, which contracts, and then sends the blood into the aortic semilunar valve that's through that valve into the aorta. Now, oddly enough, the digital description of this, as I began this a long time ago, I just found writing the digital description actually worked for me better. And I don't know why. Just strange, I suppose. But the picture there on page 343 is depicting what I just described for you in words. However, which way you get it, that's all I'm interested in is for you to understand the flow and why those things are necessary. Again, heart doing two things. I mean, most of the time, I won't include you, but I conceive just in my mind's eye of the heart is sending blood all over the body. But I forget that vital aspect of sending it to the lungs first, all right, to be oxygenated and to let the carbon dioxide out, that horrible pollutant that you spew into the air every day, causing mortal climate change. Yeah, right. But anyway, that important aspect of blood flow, you just can't forget that. So veins carry oxygenated blood in the pulmonary circulation. Arteries carry deoxygenated blood in that process. Interesting, isn't it? So again, our tendency to think the opposite with regards to those two things. That's why the real definition of arteries is blood away from the heart. The definition of veins is blood to the heart, whether it's oxygenated or not. He says, deep sigh. Yeah, exactly. And I think we're going to make it, but I'm not going to have a lot of time to check your homework, I'm sorry to say, but we'll get there. I got to collect it. I got to do it. And there's not many of you. I still might make it. Let's hustle, but I still got quite a bit of notes here. Let's see what I got. No, not that much. We're doing good. Okay, the cardiac muscle and cardiac cycle. So that's on page 345. And we'll talk about some important aspects here. Cardiac muscle is striated. It's kind of like skeletal muscle. In other words, it's got stripes. It's involuntary, like smooth muscle, and it's made up of elongated branching cells which have, and that should be an H in there, which have one or two nuclei. The cells are connected by intercalated discs that conduct action potentials from one cell to another and are self-stipulatory or self-generated by bundles of cardiac muscle nodal tissue. And there's two clumps of that. And I don't believe there is a depiction of that. There is not, sadly. So, the sinoatrial node, the SA node, is on the upper portion of the right atrium, and it's known as the pacemaker, initiating the action potentials that cause the atria to contract and stimulate action potentials on the atrioventricular node, the AV node, located near the tricuspid valve, which sends action potentials to the atrioventricular bundles, that's bundles of nodal tissue that travel through the interventricular septum and branch into each ventricle to the Purkinje fibers, which send action potentials for the ventricles to contract. So that's a mouthful, and I don't know of any way, it is not depicted here. You just got to sort of follow that through. That's the best thing I can tell you in terms of the process of the contraction. So the cardiac cycle, as you well know, because you've felt your pulse, you've listened to your heart maybe in a stethoscope, or the doctor lets you listen. But it occurs in two phases. The systolic phase, systole means to contract, is the phase of the cardiac cycle in which the ventricles contract. The diastolic phase is, well, diastole means to relax, and that's the phase of the cardiac cycle in which the ventricles relax. So you've got ventricles contracting and relaxing, and so one complete cycle then is a round of systolic and diastolic. And blood pressure is measured and referenced by those two numbers, as you well know, the higher number and the lower number. The systolic is the first number during contraction, and as you can tell, if it's squeezing and contracting, you're going to have more pressure, it's max pressure, and so that's the higher number. The diastolic is the second number, and that's at minimum pressure, we just said, and the lower number, so it goes lub-dum, lub-dum. All right, that's what you're hearing. You're hearing the contraction of the ventricles and the relaxation. The lub-dub hard side. The lub is the long, deep, booming closing of the atrioventricular valves, while the dup is the short, snapping sound of the semilunar valves in terms of those closing. So again, in terms of what you're listening for, the process that involves the squeezing is the higher pressure, obviously, and the relaxation is the lower pressure. So let me summarize then for you the last little bit here in terms of this. On page 6, veins and arteries are composed of 3 tunics. Well, actually, that is depicted for you on page 346. The tunica intima. Intima obviously means intimate, and it's the closest to the blood. So you see the tunic intima described there. The tunica media is in between the intima and the tunica adventia, which is the outer layer, and all of those are depicted in that handy-dandy little figure 1113 on page 346. The veins have thinner walls because, as you can obviously intuit, blood flows through them at a much less pressure. The vasa vasorum is a set of blood vessels that supply blood vessel walls with oxygen and nutrients. Capillaries have no tunics, only endothelium and basement membrane to allow the exchange of gases and nutrients between the tissues that they run through and the blood. Arterioles are the smallest arteries that still have 3 tunics. Venules are small veins. They do not have 3 tunics, but they only have an endothelium and a basement membrane and a few small, smooth muscle cells. So the flow in terms of this general depiction is arteries, arterioles, capillaries, venules, and veins. So heart pumping does not pull blood back to the heart. Maybe you think of it that way in terms of the heart squeezing and pulling blood back up. No, pressure pushes the blood along. As the blood travels towards the capillaries, the pressure drops, of course, but the blood gets help back to the heart by movements of the adjacent skeletal muscles which squeeze the veins and help push the blood along. So if you sit in especially a hard chair, in the same position, especially such that it cuts off, it puts pressure on the bottom of your legs, above the knees, posterior in terms of your legs. You notice your feet go to sleep. And so it's because, of course, there's a bit of pressure, maybe cutting off circulation to your feet, but also the lack of movement in terms of moving the blood back up to your heart, which, again, skeletal muscles are facilitating that movement by adding additional pressure. In the average adult, there's 90,000 miles. You think about that. 90,000 miles worth of arteries, veins, arterioles, venules, and capillaries. Isn't that cool? Isn't that cool? All right, that's all I want you to know. There's other information here which the depiction of major arteries and veins on 348 is interesting, but I'm not going to require it of you. It certainly gives the list of veins and arteries and these sorts of things, but I won't require that of you on your test. Is there any questions about that stuff? Anything at all?